Hello, my name is Christopher Cervantes, and I am currently a biochemistry graduate student at UT Southwestern. And today I'm going to talk to you all about cancer drug discovery. As you may know, cancer arises from the transformation of normal cells into cancer cells due to mutations in two classes of genes called tumor suppressors and proto-oncogenes. You can think of tumor suppressors as analogously acting like a car brake in the sense that they help to reduce normal cell growth, whereas you can think of proto-oncogenes as analogously acting like a gas pedal or an accelerator in a car in the sense that they promote normal cell growth. However, if you have mutations that disturb the functions of these genes in normal cells, that can lead to cancer cell overgrowth, ultimately leading to visible tumors in different organs of the body. And so in order to defeat cancer, both clinicians and researchers have looked for toxins that are able to stop this cancer cell overgrowth. And as a famous example here, I am showing Taxol, which was a natural product and toxin that was first isolated by researchers from the U.S. Department of Agriculture back in 1962 from this Pacific yew bark, which is shown on the right. And the way that Taxol, also known by its trade name paclitaxel, works to kill cancer cells is that it inhibits the assembly of mitotic spindles during cell division, as shown on the lower left. And this prevents the segregation of chromosomes, which is a normal part of this cell division pathway, and that is shown on the lower right. And by blocking this prevention of chromosomes segregation, it leads to cancer cell death because the cancers are no longer able to divide properly. And so while Taxol is very effective at killing cancer cells, it unfortunately is also effective at killing other normal cell types that are rapidly dividing in our bodies all the time, such as hair, intestine, and skin. And this can lead to a host of unpleasant symptoms for the cancer patient, such as hair loss, irritation to the gut, which can affect appetite, as well as rashes on the skin. And this has prompted both scientists and clinicians to begin asking, how can we discover better and newer cancer drugs? And in order to do this, our lab uses one approach our lab does use is as a phoregenetic screen, which is shown here. And the way that it begins is you first start off with a mutagenized cancer cell population, meaning, we introduce random mutations into the genome of cancer cells. And I've color-coded three single cancer cells that basically contain mutated genes that confer resistance to a potential cancer drug, as I've shown on the top here, because after we mutagenize these cells, we then select with a lethal dose of compound, and only those cells that contain mutations in resistance genes will be able to survive that lethal dose selection period. And then we can then collect those cells, harvest the genomic DNA from those cells, and send it out for whole exome sequencing, which allows us to identify common mutations in genes that we suspect of conferring resistance to the compound. And in order for a phoregenetic screen to be successful, it often needs to fulfill these different characteristics, such as it needs to be, number one, feasible, meaning the technology must exist or be invented in order for us to carry out the phoregenetic screen, all the way from introducing the random mutations into the cell population to the point of identifying resistance genes. Secondly, it needs to be an affordable process, meaning, it needs to be inexpensive, because we're screening millions of cells in order to identify those resistance genes. And lastly, it needs to be fast. And so as an example here on the right, I'm showing a common piece of equipment that our lab and other cancer drug labs use, which is called a liquid handler. And the way that this works is it's able to suck out media as well as put back in media that may contain cells. And why this is important is it allows us to both accurately and consistently plate cells into these small 96 well dishes shown um, as being these white plates here. And this allows us to screen millions and millions of cells at a time while minimizing any waste. And because the 4 genetic screens can last anywhere from 2 to 4 weeks, we need to be able to minimize contamination, but also have a great ton of accuracy. And these robots help us to achieve those goals. And this changing of media and cells only takes about a couple of minutes. And now, as shown on the right, it's able to put back the lid onto the original cell plate that it was processing, and now start processing a new 96 well plate for cell seeding. And so I hope you enjoyed my talk and learned some new and exciting things. And the last take-home messages I would like to leave you all with is that, again, cancer arises from mutations in genes that control cell growth and division. Traditional chemotherapy drugs like Taxol are very effective at killing cancer cells, but are not selective. For genetics, however, is one strategy that we can use to identify new and selective cancer drugs. And for genetics, again, allows you to connect a phenotype, in this case, cell survival towards a lethal dose of compound, to a genotype, in this case, identifying resistance genes. Thank you so much for your attention. I hope you enjoy the rest of your time at the Dallas STEM Expo. Goodbye.